Yes, sir, you are live now.
All right. Well, a very good morning, everyone. Good morning, all the students who are joining us today for the live session of MOOC, which is the second live session for this course. I'd like to welcome our students from the Proteomics course and Proteogenomics course. Currently, I am visiting University of California at San Francisco at UCSF in California. So it's night time here, but I'm very happy to join all of you. I hope you will be able to make use of our live interaction. Today we have selected topic integrated omics technologies for COVID-19 research. Of course, you have been studying many technologies in proteomics and proteogenomics. We thought, why not today we take the theme of COVID-19 and see in which way we have been using these technologies for our research. So it will be kind of a recap of various technology for you, and you should be in a position to now see like how to do the applications from these technologies. So that's the idea for doing this live session on this thematic topic. So as you have seen, COVID-19 has really showed that how scientific advancement and research, which we have been doing over the years, can be very powerful. And different technologies from the next generation sequencing to RT-PCR to cryo-EM, various technologies, and then later on the mass spectrometer, microarrays, they have all contributed immensely towards coming with the diagnostic test, prognostic test, vaccine development, all type of therapeutics. It was not possible to combat this pandemic without the help of these technologies. So it has really showed that these instrumentation technologies and our research over the years has really paid off, although it has still been very detrimental, but still probably the impact could have been much more severe. uh, so we, with our collaborating clinicians from different hospitals at Kasturba Hospital and Jaslok Hospital and KM Hospital in Mumbai, we started a project on looking at how best to understand COVID-19 and develop some of the diagnostic and prognostic tests using proteomics, metabolomics, and multi-omic technologies. It was very challenging, as you will appreciate, to work on the COVID-19, especially in the first wave of COVID-19 in India in March, April 2020, we are talking. So all the operations were very much under the BSL-3 labs, and then even preparing the samples, moving them to the lab and reaching till mass spec was all challenge. And just pictures are just describing you those times that how people have to protect themselves along with doing research. Um, I'll just kind of quickly show you this lab video, which is uh, showing me describing various instrumentation what we have in the lab, and that will help you to get at least a glimpse of our lab resources and lab technologies. And then today's session will be following with that. So just give me a second. Voice will be a bit odd here in the sense that we are talking from the live from the lab, so you will have a lot of interference background, but I think it will be good to get the feel of working in the live lab of mass spectrometers. Okay, what we are planning nearby is one of the next generation systems. And I will explain you shortly that when this transformed to virus, started showing its impact and COVID-19 was supposed to be a pandemic, we first needed to know about the sequence of this virus. And that's where the next generation sequencer and these kind of technologies are very helpful. People to genome and tell us that what is the genomic composition of the given organism which can be utilized further detect that importance. So, what can be detect the viral peptide and whatever responses are happening because of the virus to human would respond with mass spectrometry. So first of all, we try to look for how all proteins are present in the virus within the mass spectrometer. And then the goal was to look at also the host proteins which are getting modulated in this instrument. So this instrument is known as LCMS.
The first part is the electrochromatography or the analysis. And then we have electricity ionization. Then we have trying to advance spectrometer, which is one of the Ohmic technology coupled with water code and ion trap. So here the goal is to have very high resolution mass spectrometer. Then we try to separate the complex protein mixture, and that comes under deep liquid investigation. So we looked at a variety of samples from the tissues, from the endocrine swab, which is currently being used for the RT-PCR detection, as well as the plasma samples, which gives the good pathophysiological information from the patients. And these samples were deep profiled with the short-term proteomics using the method. So after the deep proteome profiling is done, the next goal is can we develop some of the simple assays or tests using mass spectrometer, which can be utilized for activity. One for looking at the SARS-CoV-2 viral peptides that is in some way are subjected to the viral peptide detection. Second, it could be when we look into the growth response and the protein, which could be prognostic biomarkers for the deep CDRP proteins. So this instrument is a wonderful master tool, which can be utilized to specifically look at the peptides which are showing maximum change in the response given. Or for that matter, we are looking at the infection of the virus. We are only looking at the specific viral peptides and hopefully you are causing some protein. So works in the field of proteomics, known as targeted proteomics, where we develop this kind of assays using connected reaction monitoring. And our core over there is to look at some specific transition of these peptides, which can be then monitored further to detect if there is a virus present or not. Or if we are looking at the post response, then we want to know whether a specific peptide from the host site is showing any alteration or is showing an abundance in response to the given that whenever a viral injector takes most immune response, certain metabolites also get hurt. And because can we investigate the plasma samples of COVID-19 infected patients and try to investigate which of the metabolites which are getting altered in the response to COVID-19. And again, we use another type of mass spectrometer. This is one of the OMICS is the mass spectrometer. And along with that, we have the USPLC. Here we can have much larger volumes of samples being injected. And that sample we are looking for with very simple organic solvents like methanol extractions. We want to look at which of the metabolites which are coming from the patients. And these kinds of these just depicts that can we start looking at the regions where the metabolites are maximally getting changed. And that, of course, is quite on a competition analysis to find out which of the metabolites we are getting changed. Okay, then let me now take you to our actual molecular biology lab. And of course, I have to wear the mask now because of the COVID situation. So we will just give you a brief, virtual tour of the lab where people do sample preparation and all kind of molecular biology based work. Of course, you're going to look at a variety of fancy instruments, but something as simple as a thermocycler that itself also is very crucial. And at the time of pandemic, that really served this purpose for looking at how viral amplification can be detected from the mRNA using real-time PCR. So again, some of the basic technology which we talk in the course are routinely used in the molecular biology lab, and that have really helped during this pandemic time. I'll take you to another instrument which is quite helpful. And let me now show you another technology which is microarrays, and we are talking about protein microarrays here. So these are high density array which is having thousands of protein printed on the small microscopic glass slide, and this actually contains almost 40,000 features consisting of 18,000 plus proteins. So now think about on these kind of high density chips with very small volume of patient samples, you can screen different type of biomarkers looking for autoantibodies or various type of protein interactors, right? So that's where I think, you can think about with modern technologies, how looking at interaction and interactomics have become quite feasible. So on the screen, what I'm showing you here is one of the slides showing the protein expression with the GST tag, and that shows that we have to do a lot of placing the grid, etc., to make sure that we can find out the right intensities of these proteins, and then that could be utilized for subsequent big data analysis. This is a small scanner which is quite helpful for scanning these chips and different wavelengths. Using one of the instruments which is not so commonly used in proteomics, but especially for the COVID investigation, we look at FTIR-based detection. Can we look at simplified methods to detect virus very quickly? And that's where FTIR instrument was quite helpful. Of course, for many of the things which we talk in the proteomics or genomics or a big data field, you need to prepare thousands and thousands of samples, right? And that's where having this kind of liquid handler becomes very handy, so that you can prepare samples in 96 well-plate format, and you can prepare them quite in the robotic manner, and you don't have to rely on manual way of handling the pipettes. So again, some of these things can be helpful if you are looking at proteome-wide or genome-wide investigation. So in the clinical proteomics or clinical omics field, you are heavily relying on large number of clinical samples, right? And that's where the sample storage becomes quite challenging. I'm sure while discussing about COVID, you have heard about the storage issue of different vaccines, especially the RNA-based vaccines, which is Moderna and Pfizer they made, and storing them at minus 80 degrees. Those kind of temperature can be very challenging. Now, what I'm standing here is in front of one of the freezer, which is having minus 86 degree temperature. And all of our clinical samples and different type of clones which are required for doing these kind of research are actually stored in that kind of condition. So you can imagine that to really prevent any kind of degradation of the samples and any contamination, we want to really store them to that level of freezing condition, right? And that's where doing this kind of research becomes quite challenging and resource intensive as well. But finally, let me show you that this is one of the big data processing server, where you can now take huge amount of data sets which we acquire from these instruments of mass spectrometers or NGS or PCR, and then we try to investigate that how to combine the data sets and look at it in a much more holistic manner.
So after giving you the lab tour, I think now I'll just now quickly jump into the exact session for today, where we are going to give you glimpses of the steps involved in doing this COVID-19 research. So when we started to first investigate this problem, we had to think about safety aspect and also thinking about how to prepare our samples for doing the proteomics work. So we tried different type of organic solvents and to inactivate virus, and then from there we took the protein which we made lysates with the urea lysis buffer and try to digest the protein, and followed by desalting and peptide estimate. Then we injected those into the liquid chromatography, followed by orbitrap fusion based mass spectrometers. So this entire workflow then revealed the entire global proteome of the nasopharyngeal swab sample, which essentially contains all the human protein and few viral peptide if there is any trace of the virus. Then next goal was, could we now try to detect those viral peptide alone and try to come up with some test for diagnosis. So in the process of doing so, we also realized that we can even take both swab sample and plasma sample to estimate the what is happening in the host. So what kind of host response we can see, like in response to the viral infection, what immune system is showing change, and which proteins from the swab and the plasma is also showing the further changes. So then we perform both, the host proteome analysis of the swab samples, as well as we took the plasma sample, which we did depletion of abundant proteins, and we further proceeded for doing the proteomics with the same workflow. So these are the some of the key steps or the broad overview of the technologies. Let me now invite the TAs to show you few key steps and the software analysis from the lab directly. So requesting Medha and Shashwati to take over now.
Good morning, everyone. Thank you, Professor Sanjeeva. So I'll be, so right now, as Professor has already said, we'll be discussing the proteomics part of the sample processing. So I'm just sharing one PPT here. So as Sir has already mentioned, we received the samples from COVID patients, and they are usually kept in a biosafety facility. So it can be plasma, it can be nasopharyngeal swab, anything. And then we process it to be run on a mass spectrometer and further whatever analysis is done. So the major steps that involved in the sample preparation for the mass spec analysis are that the sample is collected, and in case of plasma, that is what we are discussing today, the plasma is first depleted with the use of special depletion columns, and then the sample is run on an SDS plate gel to check for the quality. After which the proteins are digested using trypsin, and once the digestion is done, the peptides are desalted and made ready for a run on a LC-MS platform. So moving to the sample prep part, first, since the samples cannot be kept in the normal lab area, we process them in a a biosafety facility. So I'll be showing you one video where the sample is being taken and started with the sample processing. So here we can see that the plasma sample collected from the patients is being aliquoted and taken further for the processing. This is done in a sterile environment. So after this, we can see here that the plasma depletion column, this is a plasma depletion column. There is a resin in the plasma depletion column. So once the plasma is taken, the sample, this is the sample from the previous step, we put it in the plasma depletion column, and it is mixed with the resin thoroughly and then kept on a circular rotator for 1 hour. So this is also carried out in a sterile environment. So the steps after this, Akansha will be showing live on from lab right now. So over to Akansha.
Right, so as Megha has shown, this is the depletion columns that we have. We want to deplete the 14 abundant proteins from plasma samples. So since we are working with COVID samples and they can't be accessed like this in open, we have already done for you. And one of the depletion column with the sample is kept for the 1 hour rotation. So it has to be kept ensuring that there is end-to-end mixing of the sample for 1 hour. It's almost 1 hour. So I'll just shut this router and take out the sample. So as you can see, this is the sample. There is some turbidity, so which is a slurry. And now this sample will be centrifuged so that whatever elute comes out will be the depleted plasma, and whatever stays inside will be the slurry with all the immunoglobulins, albumins that we don't want in our sample. The next step is centrifugate this at 1000 G. So we have already centrifuged some sample for you. The next step would be because this sample is almost 300 microliter, we'll have to concentrate it. So we'll almost make up the volume to 100 microliter. Then the sample will be quantified for the protein quantification. We go for the Bradford method of quantification. And after that, that would be taken up for digestion. Once the QC is done, the QC check is done using SDS PET gel. where we see the profile of the sample, and we have to see that there is no degradation taking place in the sample when we are processing it. So next, Shashwati will show you the digestion process.
Good morning, everyone. So as you have already seen how to process the plasma sample, now I will show how I will do the digestion of the plasma sample. So here I am taking 20 mg of protein for the digestion purpose. So first I'll add TCP in there. So TCP will help in the reduction of the sample. So after adding TCP in our sample, we'll gently vortex it. For 1 hour, we will keep it in 37 degrees incubator. So this is a shaker incubator. So we have maintained it in 37 degrees. So we'll then rotate it to 300 RPM. So after for 1 hour, we'll keep it in the 37-degree incubator. After 1 hour, we will take it out. So I'll just show the next step. So we have already prepared that. So we'll now add IAA to it. So IA will alkalite the sample. So we'll prepare the IA in the dark tube because it's light sensitive. So after adding IA, we will incubate the tube in dark for 15 minutes in the room temperature. After the IA step, we will add trypsin to the sample for the digestion. Trypsin will be added in 1:30 ratio. Your protein is 30 mg, then you have to add 1 of trypsin. So the digestion will be done for overnight. So for digestion, we will keep this tube, the incubator, for 37 degree overnight, like around 16 hours. So after the digestion is done for overnight, we will take that tube for speed vac. So I'll show you. Here is the vacuum dryer. So we'll keep it open and then after 1 or 2 hours, the sample will dry, and after that, we'll take that dried sample for desalting, and then we can use it for mass spec analysis. So already we have prepared one vial for you for the mass spec analysis. So I'll take this for the mass spec analysis.
So these steps do take some time, but we have kept the sample ready for you. And now we'll be showing the sample submission in LCMSMS. So Surbi, please take ahead.
So this is the analysis. So I'll get the sample now. Right. So this we put this wire into nanochemistry. The nanochemistry, as you can see this screen here. These are the solvents. So this is the tray, and we put the wire in here, and we note the position. So it is in the D row and 8th column. You can see here what all steps are going on. So there are different steps starting from pickup of the sample, then loading of the sample, equilibrating the 3-column, equilibrating the analytical column. So this nano LC is connected to the mass spectrometer, which is an orbit trap mass spectrometer. And these are the
nano wipers that connect the nano HC to the mass spec. This is the P column and this is the analytical column. So the sample is loaded onto the analytical column, and then over a gradient of 1 or 2 hours, depending on the sample complexity, the peptides are diluted, which goes inside the data. Please let me know once the screen is shared. This is the interface.
Yes, it is shared.
Where we line up the samples, as you can see, here putting the details, for example, the sample type, the file name, the path where we want to save the raw file, the instrument method, and the position. So here we start the run, and then this is another software where we observe how the chromatogram has come. So just for representation, this is one chromatogram for a complex sample like plasma, and this is how a chromatogram looks like. You can also zoom into the chromatogram to see the MSMS, to see the MS and MSMS, and this is how the zoomed in chromatogram looks like. Thank you.
Thank you, Surbhi. So this is how the LC-MS analysis is done. And we get the raw files from here, which are further analyzed on particular identification software, which can be any type of software, like open access MaxQuant, or more proprietary softwares like Proteome Discoverer. So now moving on to the next step, that is, once we have done the LFQ analysis, we move on to the targeted analysis. So basically from the discovery data set, we have a few differentially expressed proteins which are coming significant in the samples. For example, in one of our previous studies for COVID-19 samples only, plasma samples, we could differentiate the samples between severe and non-severe based on a few of the proteins that we were seeing. So example like APO2 and other like CFP, serpinase6. So you can see that the levels are differentiating here. This is just an exemplary figure, illustrative figure for the purpose here. So now we have these proteins are in our hand, and we want to see like whether they will have any actual clinical importance. So we take a few of the proteins and do an targeted proteomic assay. So there are many kinds of targeted proteomic assays, but today we are going to focus on MRM. And the MRM study, basically we use a triple quadrupole mass spectrometer, which has in which particular peptides and transitions are selected, and data for them is acquired, and the data analysis is basically done on a software like Skyline. So I'll be just showing you how we proceed with the experiment. So this is Skyline. So before starting with the targeted experiment, you come to the Skyline software and you prepare a transition list that needs to be given, like fed into the instrument. So this is a blank Skyline document, and I need to input here the protein ID that for which I need to prepare the transition list. So this list needs to be given to the instrument. So I'll go to UniProt and I'll search for the protein of interest. So for example, in our case, if I'm looking for fibrinogen. So it's searching. So we are looking for, so I'm more interested in, I'll just type it again, FGG protein. So this one is that. So once you click on this, this particular page will open. So you know the UniProt ID from here. I can open here and file, edit, insert protein. So I can just input the UniProt ID. I'll copy it from here and input it here. Okay. So basically we have to check for a few settings also, and you have to check the background proteome. This is available in the software. So once we do that, our protein will come here. So now you can see the protein is being named, and all the peptides that are coming for that particular protein you can see here. Now you just have to export the list, the transition list, based on the instrument that you're using, and you can click OK, and then you'll have the transition list. The transition list will be taken ahead, and now we'll go to the triple quadrupole mass spectrometer, where the transition list will be, like the sample will be run according to the transition list. So over to Shashwati right now.
Okay, so now over to Shalini. She will explain the Metabolomics experiment, how we are planning the experiment and all that.
Hi all. As you
Shalini,
remember,
if I just interrupt you for a minute before you
okay, sure.
start. So let me just give them the glimpse of the results. If I could just share my screen. So after doing the experiment which Medha had described, what we actually tried to achieve from that is look at which peptide we can select for targeted screening, right? So first question we had was, can we detect even virus from the nasopharyngeal swab sample? And we then selected 11 peptides from... Am I audible?
So sorry, sir. Yes, you can go ahead. Actually, you were not audible earlier. That's why. Now it's audible.
So we selected 11 peptides from the spike protein, nucleoprotein, and replicase polyprotein to cover different proteins of virus. And then the workflow which Medha described, we use those 11 peptides for with the 8 minutes of elution to look for the viral detection assay development. And likewise, from the plasma sample, we tried to look at which proteins can be used to determine the severity of the disease. And that's where we looked for the non-severe and the severe form of disease. And as you can see from the plasma, we could see very different intensity levels from the non-severe and severe, which is very helpful to tell an individual is going to be more prone to severity, right? And that was the kind of question which we needed to answer that time, that among so many individuals, which one require more clinical interventions. And then as a follow-up of this work, we also were given some samples by the clinicians to look into how the long-term COVID impact will be there on human reproduction system. And then these were the men who were having COVID-19, but they recovered, and their semen samples were analyzed, which we then could see that, okay, some of the very key essential protein for spermatogenesis and sperm motility, even their levels were still quite down, and like protein like seminogenin protein, multiple peptides of those are showing the down regulation. It means that individuals have not completely recovered. And we also looked at some other proteins of the host, which are very critical for the male reproduction system. So what it conveys you that, variety of tests can be developed with these technologies. Workflow remains same. You have to only look for which proteins is of your interest, which you can use for further testing. So we are just trying to utilize these workflows to come up with various type of assay development. And that was a part of the targeted proteomics. Now then, idea was from the same plasma sample, could we also try to understand what all changes happens in the overall because of viral response, and can we integrate that data, multi-omics data, to obtain the mechanistic insight of the COVID-19? So that's what now the next segment will be from Shalini and team to talk about metabolomics and integrated omic analysis. So Shalini, now you can share your screen.
Thank you, sir. So now I'll share my screen. So as you remember, as Professor Sanjeeva just now explained the importance of viral inactivation, but there is difference between a proteomic sample preparation and during that when you are inactivating the viruses, and when you are inactivating the viruses for the metabolomic samples, because as you know, metabolites are highly dynamic and very sensitive. They switch to the different molecule very quickly, and that's why we need to have a method which do not disturb the metabolites, but it will keep only it will only degrade the COVID virus. So now there are two different type of methodologies: physical methods and chemical methods, and out of which physical method includes heat and UV, whereas chemical methods includes detergent, alcohol, and pH, out of which for us alcohol worked best, and we had used alcohol for inactivation of the viruses, and then followed by air drying in lab. So after this whole process was done in the place where the whole unit was, it was only for COVID, and it was not done in the laboratory. That's why we are not able to show you here. And next step after this is the sample preparation, where the plasma sample is taken and the whole procedure is done. So we'll not explain you here much about it, but we'll show you how exactly in the lab, how this is done. So over to Nirjar now, who will show you how the sample preparation is done and what is the importance of QC pool and internal standards. So over to Nirjar now. Thank you.
Hello everyone. So let's start with the very basic methods of sample preparation. So sample preparation depends upon many factors: the type of samples, the type of metabolites that you want to find. So just starting with it, serum samples are one of the most common type of samples that we do metabolomics with. So here that I'm showing is one method, which is 1:4 sample is to methanol. So we add 4 times of chilled methanol, and then we incubate it. So basically what methanol does, it takes out all the small molecules into it, and when we after that centrifuge it, it comes into that supernatant of methanol. So as I first mentioned, that there are several methods of extraction, depends upon which type of samples we want. Methanol extraction is one of the most global methods that we can have. So right now we added 50 microliter of the samples, and to it 4 times, let's say 200 microliter in that case of methanol. This methanol has to be chilled. So being chilled, it creates an instant separation of the metabolites from the other compounds that we have in the cell. So let me just show you once. It looks, it will create a foggy atmosphere, and after that you can understand that there will be some precipitation on it. So based on the concentration of the serum sample, there would be separation of various other debris. So as I was previously mentioning, there are several methods. Chloroform methanol is another one, a combination which gives both hydrophilic as well as hydrophobic metabolites. So metabolites is just a threshold for size. So there are various type of molecules that comes under the word metabolites. It can be very small peptides or amino acid. It can be lipid molecules, carbohydrate molecules. So based on that, whoever wants to extract what kind of metabolites, they have to choose that kind of extraction strategies, and so on and so forth with the columns. So columns are also there: C18, helic, C18 for the hydrophobic, helic for the hydrophilic, and amide for the in-betweens, and so is the solvent system. So anyways, coming back to this extraction procedure. So after getting the chilled methanol added, we would put into the -20 degree incubator. So this -20 incubation can be kept for half an hour, for overnight, based on whatever strategies the person that has optimized it with. Some do it quick, whose concentration of the sample is very high. Some do it overnight. Well, overnight reaches a saturation of the extraction that can be possible from the methanol. So as we are going around with, we cannot show the overnight incubation right now. So we are showing an example of how it would look. So after that, we have to do the centrifugation that we get out of the incubation and separate out the supernatant. We don't need the pellets. So the pellets that we would find after the incubation are basically the protein debris, cell debris, etc. Not cell in case of serum, but the other debris other than methanol. other than metabolites. So after that, after the centrifugation, we would get what we call as the extracted metabolites in methanol. So one thing, sorry, I missed. During the sample, we also have to add internal standards. Sorry, my bad. So we generally have multiple internal standards. So based on how much you want to do targeted, will have separate set. If you have targeted, you have to do, let's say, labeled metabolites as standards. Or if you're doing just a global study, a subset of standards can be added to the samples. So just like we add synaptic acid during just when we added the sample, and then after we take into the centrifugation, we have to do the speed vac. The speed vac, what it would do, it would concentrate the sample, and then after speed vacuum, we would reconstitute it in 50% methanol, because that's how our solvent system and the C18 works. And to it, we would add another the post extraction what we call it internal standard. In this case, we add reserpine in our workflows that we have done. And this will also help us to understand the differences of the instrumentation variation, because internal standards are added, spike in standards, rather to say, are added to understand the internal variations, the instrumentation variations, so that the sample, the alterations that we are having are not into the other variations other than what the sample, the biological data means to it. So after that, we put into the MS files like these, and we submit into the instrumentation as similar as you saw in the previous case. So that's it from here. Moving forward, the analysis and the methodology in the MS will be shown by Prof.
Thanks, Nirjit. So as Nirjit, my colleague Nirjit told you about the sample preparation part, now we'll go for the sample processing part. So in the mass spectrometry. So once the samples are submitted to a mass spectrometry, so here is the Excel sheet where you can prepare write the sample names date wise, and according to you have to mention the position of the sample where you're keeping the vials in the mass spectrometry. So once you are done with this Excel sheet where you are giving the information about the samples, your QC pools, your the blanks and the unknown samples, and along with the injection volume that you have to optimize for the mass spectrometry for different samples, then you have what you will do. Then you have to import the sequence. So we'll close the sequence from here. So then after that we have to import the sequence into the Excalibur. So it's a Thermos software. So where we run the samples. So based on the location of your file in the system, you have to load the samples. So here the software will ask you about so many options. So whatever you have to, the important relevant options are as per the software that you have to give. Like we have to give this injection volume, like the position of the sample, no need of processing method, and the instrumentation path, no need of sample ID. We have to give the file name, and we have to give the sample type, and then you have to press OK. Now you can see that the sample list is loaded into the software. You just have to copy all the samples, and you just have to press the run button so that the all sequence gets started. So after the samples they are over, then comes the analysis part. So as Nirjit told you about the pre-spiking and the post-spiking. So basically for the metabolomics part, we do the pre-spiking with the synaptic acid. So here you can see my screen on the middle one. It's basically a reserpine, so which is basically a post-spiking. So in both positive as well as in the negative mode. So here is on the upper chromatogram show the TIC, which is basically the total ionic current, which is basically show the spectra of the all the metabolites all together. So this is basically a QC sample. So one thing that you have to keep in mind while analyzing the data, that the RT of the internal standard should be the same. Now you can see it's 9.53, 9.60, and the 7.92. So this is 9.53 is basically for the reserpine in the positive mode, and 7.92 is basically for the synaptic acid in the positive mode. So when you check for the another sample, so this RT should be around the same. There should be no variation in the RT. So when you look for these things, you must be sure that the RT of the internal standards, both synaptic acid as well as for the reserpine acid, should be same throughout. And this is the total ion current. So this is the actual file of the metabolites which we got after running the sample. So after this file, we have to detect the software, this metabolites, which we basically go ahead with the compound discovery software, which is a proprietary software from the Thermo. So where we can get the metabolites and we proceed downstream with the analysis part. So now my colleague Ankit Haldar will show you about the analysis part. Over to you, Ankit.
Yeah. Thank you, Gautam. So now once you get the analysis and do the secondary analysis using different statistical tests like t-test pass metabolites or any other two other statistical tests. So now you will get a list of the proteins and as well as the metabolites. So for proteins, you will find the differentially expressed proteins, and for the metabolites, you will find the differentially expressed metabolites. So now you will get different kinds of list like this, where you will find the UniProt IDs of the proteins from the MaxQuant and after analyzing it in statistical tests, and you will see the alteration as the upregulation or the downregulation the different proteins, and as well as you will get the different metabolites, the different metabolites which are being down regulated as well as upregulated. Now you want to understand that what are the pathways that they are getting affected due to the salted proteins and the metabolites. So for example, so you will get an UniProt ID. So using UniProt, you can just convert these UniProt IDs. I will just show you a demo. So for example, you can take the UniProt IDs, and if you just place it in UniProt, so you can just from here you can get to gene name. So here in this way you can just convert the UniProt and the gene names, whereas in the metabolites, for the metabolites, there are different IDs that can be used, like HMDB ID, that is the Human Metabolome Database ID, PubChem ID, or the KEGG ID. So for the pathways, generally KEGG ID, as we want to know the KEGG pathway. So what we will do is that for metabolomics translation to the different IDs, so you can just copy the names of the different metabolites, and there is a like chemical translation service where you can just do a batch conversion. You just can click it, and here you can paste the IDs, and from you can provide the options like chemical name to convert into KEGG or Chevy or HMDB IDs, and it will just get converted. So here we have made the list of the different gene IDs, UniProt IDs, and the KEGG IDs. Now we will open a software which is known as Cytoscape. So here in this Cytoscape, we will install a plugin which is known as a Medscape. So in the Medscape, it gives us it can provide the comprehensive network with both integrated proteomics and metabolomics data. So whatever proteomics data and the metabolomics data that we have analyzed, we have found that differentially regulated proteins and the metabolites in different cohorts of the severity, non-severe COVID groups. So we can now see the pathways that are actually getting altered or affected. So in this plugin, we will add the compound IDs. So here it takes the KEGG IDs. So you can see that it is written that enter a list of compound names of the KEGG IDs. So we will put the KEGG IDs. So I have already put the KEGG IDs list that we have got, and here we will put the gene IDs, that is a differentially regulated proteins. Now once we are done with it, now what we will do is that we will use the compound genes. So here there are different types of pathways that we can build. So as you know that our metabolic pathways are related, that one compound reacts with another compound, and it is catalyzed mostly by an enzyme which is regulated by different gene expression. So we can give as compound reaction enzyme gene, or we can give as compound and the reaction, that what is the compounds and what is the immediate reaction, or we can give a correlation between a gene and a compound, or we can give only compound. So first we will see that what happens if we give compound reaction enzyme gene. So here we can see that so you can see that a complex network has been built up using compound, where red defines your compound, blue defines your protein, green defines your enzymes, and the reaction. So everything is mentioned here. So red, blue in different colored annotations it is provided. Now if we want to see what are the pathways, so you can see that amino acid sugars metabolism. So here all the both the genes as well as the metabolites have been taken into account, and the whole picture of what is happening inside the body due to the infection is shown here. Then androgen and estrogen biosynthesis and metabolism, fructose and mannose metabolism. So these are all carbohydrate metabolism, galactose metabolisms. So we can get a glimpse of what are the different pathways which are getting affected due to it, and from here we can try to predict what is the reason because of the severity is driving forward. Now if we go for the compound reaction, then also we will get different like the same metabolisms, and here only the compound and the reaction is given, and all those genes and enzymes have been waved off, and you can now dig deep into understand what are the compounds and what are those reactions that are actually taking place. Now if we go for the compound gene, so you will see that, so these are the compounds and the genes. So which genes are acting on which pathways and with which expression their compounds are getting altered, you can get from here. So the pathways will remain the same, but you can go deep into it and can understand its various at its various pathways and its various reactions that are actually getting altered. And finally, if you do give just compounds, so you will get that only the compounds and their interactions, like you can see that L-ornithine is hydrogen. So these red, dark red are those compounds that are acts as a master regulator and are regulating different are linking different reactions, and for the alteration of this particular compound lead to alteration of other different pathways. So these are the things with which comprehensive starting from the comprehensive sample preparation, then after analyzing the data, we can get the comprehensive view of what is happening actually in the disease. So this is all from my side. So thank you.
Thank you, Ankit. So as he has explained all the information about how to use a Metascape plugin in the Cytoscape. So this is one of the things what we had used in one of the papers, which Ankit had taken as reference. And we were able to find a different pathway which was not coming up as such with the proteomics data or as metabolomics data. But when we try to compile these two lists together for metadata analysis for using all the literature background findings from proteomics as well as metabolomics papers till date, we were able to find a novel pathway which was related to the reproductive health, affecting reproductive health of males. And later on, we were able to see multiple manuscripts coming, research papers coming up, saying the same thing and confirming that what we had found using the metadata analysis was actually right. So,
Actually, so thank you
so
and
what
the
do
TL of Metabolomics to explain the workflow. I'll slightly interrupt you now and try to come up with a concluding session. So what you have seen, like of course the ease of doing this analysis you have seen. It requires a lot of practice, which I'm sure you will do that. I'll just take one question from the portal first from Ayushi. I think she asked about the protocols of different samples when we are analyzing of irrespective of their age or their variations. Are we using still same protocols? So first of all, Ayushi, like when we do clinical studies, we do try to keep the inclusion criteria and exclusion criteria very well defined, right? Because these studies comes with the ethical approval and IRB approval, regulatory approvals, where we have to define that what is our study group. So we do try to keep our study group as homogeneous as possible, given that you are right that there will be a lot of variation into pediatric versus the old individuals. So we keep it like in the young age above 18 to 50, 55, that kind of age group. And then we try to apply the same protocol so that we could see with the same method what could be the differential expression of the protein or metabolites, which could be mapped with our technologies. And but while analyzing the data, then of course one need to keep in mind that what was the gender, what was the age, what is the other clinical symptoms they had. For example, in this case, we looked for ARDS or pneumonia or SpO2 oxygen level. And then we also try to look into those factors into analysis, right? So again, those variations can be brought into more at the analysis level, but the sample preparation remains the same. So just coming back here, now we took the same plasma sample. Idea was, if we have individuals who are having both like mild individuals and the, the individuals with the severe form of the COVID-19, can we, see in which way the disease get progressed. So therefore, we started using different time points for the longitudinal samples of these patients to look into longitudinal analysis of using proteomics and metabolomics. This study was featured in the cover page of Journal of Proteome Research. And what we deciphered from this, that several pathways using proteomics, we could see like, myeloid leukocyte activation came as another key change which was happening because of the severity of the disease, along with platelet degranulation and the complement and coagulation cascade change. And with the metabolomics, various type of metabolites, especially the arginine metabolism, turned out to be the key regulators of fatal symptoms of severe COVID-19. So these are the various type of analysis which reveals some biology. Additionally, we thought now, since we have seen there are changes happening in the metabolic fingerprints of the mild versus severe, can we apply simpler technologies like Fourier transform, IR technology, to find out the changes between non-severe and severe patients? And we use the same plasma samples, and now we try to measure the FTIR spectra of these patients to look into the regions of the wavelength band where there could be changes from the non-severe and severe. And after analyzing this, we had a team of bioinformaticians from QAMR in Australia. They were given the blinded data set, which they could, after making the model, they could predict almost 95% sensitive test, which was being developed. And this was the cover page of the ACL journal in the analytical chemistry, which got a lot of media coverage as well. So just to summarize what we have been talking in today's live session, we thought to give you glimpses of using various proteomics and mass spectrometry and metabolomic technologies and omic based investigations to look for different problems of COVID-19, whether a detection of virus, looking at prognosis of virus, identifying the novel pathways, looking into even the long-term impact on the human reproduction, and a variety of these problems could be resolved using these technologies. And we are very happy to have invitation from Nature to give a webinar to talk about these, how these kind of technologies could be utilized to investigate these complex problems. And of course, it showed that there could be alternate approaches of, investigating these problems. So, I hope you found these course. The Proteomics students are finishing their course shortly. You found this course useful to give the glimpses of new technologies. I hope you are able to now, in a better position to decide that which technique can be useful for which type of biological problem which you want to investigate. And of course, you can now build further from here. And those who will be topper of the courses, we will give you opportunity to come and spend some time with us in our lab as interns, or interact with us, or participate in some conference or workshop with us without any additional cost. We will be very much supportive to take your knowledge to the next level of the lab exposure, right? And those who are continuing on proteogenomics, I'm sure you will have more interaction with us over the coming week. So wish you good luck for the exam for the Proteomics course students and the continuing students on proteogenomics. Wish you continued luck and interest in this subject. Let me thank all the TAs who today showed you many small steps, but those were very critical, and they managed very well within the time, along with making the different type of demonstrations directly live from the lab. I hope you found some of these session useful, and probably it will give you more interest into this subject, that how you can take these basic concepts and apply those to the real life problems. Of course, we work into the cancer and many other complex diseases as well, and the same technologies can be used for different type of investigation. So stay in touch with us, and we'll be very happy to take your online knowledge to more, kind of in-person interactions in future. Okay. Wish you good luck, everybody. We will stop the session now. Thank you very much.
I'll stop.